Hello, I hope you're all doing fine. Today's lesson is about the cells of the immune system. This is going to be activity 4, page 121. First of all, we're supposed to know how can we prepare a blood smear. First of all, of course, you're supposed to sterilize the finger you're going to prick. Then after pricking the tip of the finger, you have to place the drop of the blood on a microscopic slide, and using another slide you push the drop of blood rapidly over the first slide in order to form a very thin film of blood on that slide. Then you let dry and add alcohol to fix the smear. You then wait few minutes before adding using solution to stain the red blood cells, of course. And then you also wait for another few minutes before rinsing with water, then adding drops of methylene blue in order for you to stain the white blood cells. And then you rinse with water and let dry, and you observe under the microscope. This is how our blood smear is going to appear under the microscope. You are going to see the big number of red blood cells which have a disc shape, biconcaved structures. And you are going to also notice the presence of the white blood cells. They are less numerous, as you can see. Some of these white blood cells have a background nucleus. These are the lymphocytes. Others have multi-lobed nucleus. They are referred to as polymorphonuclear cells, and these cells are said to be the granulocytes. And others have a U-shaped nucleus, or they are known as the monocytes. You have to know first that all of the blood cells originate from the stem cells, which are known as the hematopoietic stem cells, which are present in the bone marrow. These stem cells are divided into two types, the myeloid and the lymphoid. The myeloid progenitor stem cells are the origin for the blood platelets, which are responsible for the clotting of the blood. To prevent bleeding when you have any wound or injury. Also, these myeloid progenitors are the origin of the erythrocytes. The erythrocytes are the red blood cells, which are responsible for transporting oxygen and CO2 to and from the cells. So they are responsible for transporting the respiratory gases. And myeloid progenitors are also very important stem cells that result in the formation of the granulocytes, which are of three types, known as the eosinophils, the basophils, and the neutrophils. So these three are the granulocytes. And as you can also see, the myeloid progenitors they are the origin for the monocyte population of the leukocytes as well. On the other hand, the other type of these stem cells in the bone marrow are known as the lymphoid stem cells, which give us the population of the lymphocytes. We have two types of lymphocytes, the B and the T lymphocytes. So this is a very important document. You're supposed to know that from the bone marrow stem cells we have two types, the myeloid and lymphoid, and what are the origin of every single cell that originate from each and every one of these two types. First of all, we are going to notice the shape of the nucleus and the shape in general for each of the following cells. As you can see first, the granulocytes, usually the granulocytes are of three types. These are the granulocytes. They are the eosinophils, the basophils, and the neutrophils. They have a diameter of 10 to 12 micrometer. Their nucleus, as you can see, is multi-lobed, so they do not have a regular shaped nucleus. They have instead a multi-lobed nucleus, and this is why we call them polymorphonuclear cells. They represent about 67% of all the population of the white blood cells, and they have a lifespan of 2 to 3 days only. As for the monocytes, as you can see, the monocytes have a horseshoe-shaped nucleus or a shaped nucleus. They have a diameter of 10 to 18 micrometer, and they represent only about 6% of the leukocytic population. When we say leukocyte, we mean white blood cells. They have a very important capacity of transforming to macrophages. So the monocytes become macrophages when they want to engulf and phagocyte the pathogenic agent, so any foreign body entering to the body and considered as a non-self. They have a lifespan that might range from 2 to 5, 6 days approximately. The lymphocytes are characterized by their big round nucleus. They are of smaller size if you compare them to the monocytes and the granulocytes. They have a diameter that ranges between 7 to 8 micrometer, and they represent around 25% of the leukocytic population. We also have other type of cells known as the mast cells, which look like the lymphocytes because they have a big round nucleus as well, but they are characterized by the presence of granules similar to those that you can find here. And these granules are filled with a chemical substance known as histamine, and the release of histamine is what leads to the allergic reactions for the individuals suffering from allergy. So the mast cells have receptors for certain types of antibodies, mainly antibodies immunoglobulins of type E. Types of the granulocytes. As we said before, there are three types of granulocytes, which are the neutrophils, eosinophils, and the basophils. Neutrophils are the most abundant type. They are the most numerous type among all types of granulocytes. They have a very important property in undergoing a very important process, which is known as the phagocytosis, or engulfing and eating, digesting any foreign body they might meet. And their granules can be stained by neutral dyes, and this is why we refer to them as neutrophils. As for the eosinophils, they also undergo the process of phagocytosis, but they are mainly involved in the phagocytosis of complexes made up of antibodies and antigens. So when the antibodies fix to certain antigens, it is the job of the eosinophils to digest them. They have acidic staining granules. The granules can be stained by acidic dyes. As for the basophils, they are the least abundant among all, and they are very important in inflammatory responses, and their granules can be stained by basic dyes. Regarding the monocytes, as we said before, they have a very important property, which is the property of phagocytosis. But what makes the monocytes different from the granulocytes is that the phagocytosis that the granulocytes ends as soon as the pathogenic agent is digested. As for the monocytes after undergoing the process of phagocytosis, they first become macrophages to undergo the process of phagocytosis, and after that they will express fragments of the phagocyted elements to their surface, and they will change into what we call antigen presenting cells. Let us see this process of phagocytosis. First of all, this is the macrophage. When the monocyte becomes a macrophage, it will have this structure, which is having some ridges, and this is the process of engulfing, in which the pathogenic agent becomes embedded within the pseudopods. These are said to be pseudopods or false feet that the phagocyte, in this case it is the macrophage, uses in order to engulf the pathogenic agent. As the pathogenic agent is mixed with certain enzymes, which are the lysosomes present within the phagocyte, within the macrophage, these lysosomes are going to digest the pathogenic agent. After being digested, the remnants are going to be, these leftovers or remnants are going to be released outside, but some of these remnants are going to be expressed to the surface. In this case, we call the macrophage an antigen presenting cell. So it is presenting those antigens for other types of cells, the T4 lymphocytes. We're going to discuss this actually later, in order to tell that some foreign elements are present in the body, and maybe there are more of these elements that need to trigger a specific immune response, and this is why it changes into an antigen presenting cell. This is a very important property that the macrophage undergoes. Let's move on into the lymphocyte populations. There are three different types of lymphocytes: the B lymphocytes, the T lymphocytes, and there are also the natural killer cells. They all have a big round nucleus. Regarding the B lymphocytes, the main important role that the B lymphocytes undergo is the production of a Y-shaped protein called antibody. This is why we say that the B lymphocytes are the antibody secreting cells. They are involved in a specific immune response which is targeted against the specific cells, specific antigens, and this is known as a specific humoral immune response because it's carried out by the antibodies present in the serum and not by the cell itself. And these receptors that you can see, which will appear at the surface of the B lymphocyte, is said to be a B cell receptor. They are usually, the B lymphocytes are produced and they are matured in the bone marrow. They transform into what we call plasmocytes or plasma cells when they are ready to secrete their antibodies. This is the plasma cell. When the B lymphocyte is ready to start producing the antibodies, it will be converted into plasma cells characterized by well-developed endoplasmic reticulum and of course Golgi bodies, because these are involved in the production of these proteins or the antibodies. T lymphocytes, on the other hand, are a very important part of the immune system that develop in they are formed actually in the bone marrow, but unlike the B lymphocytes, they do not mature in the bone marrow. Instead, they mature in the thymus. They are very important in the protection against certain infection, but most importantly, they can also help in the fight against cancerous cells. They have T cell receptors, TCRs, on their surface, and they are involved in cell-mediated specific immune response, not like the B lymphocytes, which are involved in the humoral-mediated specific immune response, because the B lymphocytes secrete antibodies, and these antibodies are important in doing the immune responses. For the T lymphocyte, it is the T lymphocyte itself that undergoes the immune response, and this is why we call such immune response a cell-mediated specific immune response. Among the T lymphocytes, we have two types of T lymphocytes: the T4, which are also called the T helper lymphocytes, and the T8, which are also called the T killer lymphocytes. So if you take a look into a T4 lymphocyte, you will notice that the T cell receptors on its surface are of type CD4. CD stands for cluster of differentiation of type 4. As for the T8 or T killer cell, the cluster of differentiation on its surface, CD, is said to be CD8. Mast cells. These mast cells are cells having lots of granules which are filled with histamine, as we said, and they possess receptors which allow the fixation of specific type of antibodies known as antibodies of type E or immunoglobulins of type E. Once these antibodies fix with their specific antigens on the surface of these receptors, they are going to trigger the release of this histamine, which will cause the allergic reactions. Natural killer cells are also a very important type of lymphocytes. They are important in actually non-specific immune response, unlike the lymphocytes which undergo a specific immune response. These type of cells play a very important role in the host rejection of tumors and infected cells. I hope that you're going to cover this activity very well, and I will see you later in other activities. Thank you.